Hello. So after some gap, I'm back with lecture number 10. So we have discussed some of the hybridization based techniques before, that is northern hybridization, southern hybridization, etc., and also RNA in situ hybridization. Um, Today I'll be talking about first microarray and RNA-seq kind of techniques, and then I will go back and give you precise input about which hybridization technique is used when and for what purpose. So, you will face these kind of questions often, that how would you identify genes that are expressed under a specific condition, say when its cells are grown in the presence of sucrose. So there will be a change in gene expression profile. Some genes that are not expressed when cells are grown in presence of glucose will now be expressed in the presence of sucrose, and some genes that may be expressed in presence of sucrose, may be expressed in the presence of glucose, will no longer be needed to be expressed in the presence of sucrose, and their expression will go down. So how do you recognize those genes? Um, um, if you want to know that, see, like there are these proteins called transcription factors, which are expressed in specific tissues at specific times. If you want to know that which are the genes whose expression is dependent on the presence of a specific transcription factor, how do you go about it? Or signaling pathways. If you want to know the genes that are expressed when yeast cells are treated with a pheromone, that is a mating factor, or my favorite topic, if the cells are exposed to BMP ligands, what are the genes that are expressed specifically under those conditions? Or it can be something much broader than that, genes that are expressed in one type of tissue versus the other. You can uh, compare genes that are as different as apples and oranges, like muscles versus neurons, or neurons versus skin, those kind, or tissues that are slightly different from each other. Without introducing you to the terms properly, there is a tissue in the developing embryo called perichondrium. If you want to know which of the genes that are specifically expressed in the perichondrium, or in the cartilage that lines our joint, that is the articular cartilage or joint cartilage, or in developing bone cells, depending on which kind of laboratory you are in, which kind of research problem you are addressing, you will always be faced with this challenge of identifying genes that are expressed under one set of conditions and not expressed under another set of conditions. So this is like the historically the methods have evolved several times, from rather complex ways of doing it to relatively more simpler ways of doing it. I will skip some of the earlier methodologies, and I would rather focus on the more recent methodologies. But just so that you have an appreciation of history, let me tell you a little bit about run-on transcription. So run-on transcription is, so as you know that mRNAs are transcribed in the nucleus. Okay, so if you can manage to isolate nucleus of a cell and very briefly expose it to P32 UTP, it will radiolabel all the RNA that were being produced in that cell at that particular time when you isolated the nucleus. Now, if you want to know which are these mRNAs that were being synthesized at the time, in the olden days, laboratories will have a blot of a blot that is, like some kind of a membrane on which certain known cDNAs were immobilized. Those were relatively low throughput experiments, like 15, 20, 30, 40 genes will be there, and you put the radio labeled RNA on that piece of paper, and the cDNAs which are represented and corresponding to the mRNAs that are radio labeled, they will light up, as you can see in this particular one, where if you have liver specific DNA array, then some of those genes will light up when you take the nucleus from the liver. But if you take the nucleus from kidney or brain, those will not light up, while some of these in the lower panel will always light up, because those are the housekeeping genes like tRNA or beta tubulin, alpha tubulin, etc. So those are the housekeeping genes. So that was the old-fashioned way of doing it. I'm not going to get into the subtractive hybridization or differential display, because those methods are no longer used. If you are specifically interested, I will explain it to you during one of the discussion classes, but these are not really used. The method that all of you have heard about and you have some idea about perhaps is called microarray. So what is a microarray? Microarray is just a high throughput version of this. So here, like you had a couple of dozen cDNAs in this specific example shown, only a dozen cDNAs, known cDNAs arrayed on a piece of paper. But with the improvement of lithography and inkjet printing technology, this could be miniaturized many-fold over. What do you mean by miniaturized? Meaning instead of this really large blot of paper, a regular glass slide, like 3/4 of an inch by 2-inch kind of a glass slide. On that, you can have cDNA representation of entire genome. Okay, and that is so. First, you should understand what is a microarray. So this is an array. Okay, this is an array. This is a large piece of paper on which there are only 12 cDNAs spotted. This much more miniaturized micro version is called a microarray. So you can think of, just hold on a minute, let me get my stylus. It will be a much smaller, like strange. It looks like the stylus is no longer working. Sorry, folks. So forget about it. The microarray is basically a normal glass slide that you may have seen in hospitals, in path labs, in laboratories if you have been to. On that regular glass slide, you will have spots of cDNA for thousands of genes. Now, that's what is a microarray. And what you do with a microarray, so exactly like this, but instead of radio labeled RNA, you use RNA that are fluorescently labeled. And you do not use multiple different blots or multiple different arrays. Sometimes you do. There are some technologies that are based on different arrays for different conditions. But generally speaking, you can use on the same array uh, mRNA is isolated from condition 1 and mRNA is isolated from condition 2. From condition 1, the mRNAs are fluorescently labeled with, say, green color. From condition 2, mRNAs are isolated with fluorescent label of red color. And then you use a microscope with fluorescent detection and very high resolution to see which spot is just red in color or just green in color or yellow in color after merging the two different images, so that you know that which mRNA was present both under condition 1 and under condition 2. Like, I have represented it schematically like this, that imagine this is a whole microarray and you isolated mRNAs when cells were grown under condition A, and it can be any condition, okay? Any of those that I described previously, like in presence or absence of a nutrient, or in presence or absence of a transcription factor, or in presence or absence of a signaling molecule, or different kinds of tissues, or the same cell at different points of time, whatever. Just two different conditions. The condition is defined by you, and you want to know the mRNAs that are produced in one condition and not in the other. That is the simple question that you generally ask using microarray. So say you label the mRNAs that are produced under condition A with red fluorescence. So schematically, this is how the array would look like under the microscope. And you label the mRNAs that are produced under condition B, and this is how the array will look like under the microscope. And then through image processing, there are softwares available by which you merge these two images, and you see that majority of the spots are yellow, meaning these genes were expressed both under condition A and under condition B. There are some spots that are only green, meaning those genes are exclusively expressed under condition A, and some spots that are only red, meaning those genes were expressed only under condition B. So this is how, so you replace condition A or B with presence and absence of sucrose. You get to know which are the genes, all these green ones that are expressed only when sucrose is there, and the ones in red that are normally expressed in presence of glucose but downregulated in presence of sucrose. Okay, or say you want to know the genes that are regulated by a particular transcription factor X, then the green ones are the genes that are produced when X transcription factor is present in your system, and the red ones are the ones that are normally present, but when X is there in your system, then those are no longer present. Okay, so these are different kinds of analysis that you can do. Now, what you should know about is the evolution of microarray technology, because that will allow you to appreciate where we are vis-a-vis this technology today. So the critical aspects of microarray analysis is, first, what is there on the slide? Okay, are those ESTs or oligonucleotide? This makes a whole lot of difference. So ESTs, as I told you in one of the discussion classes, is called expressed sequence tags. These are produced by reverse transcription followed by PCR, and reverse transcription of mRNAs is anchored at the poly A tail with an oligo DT, and then there are different methods to amplify it from the 5' end that we have discussed when we talked about cDNA library construction. Now, the reason these are called ESTs is because the vendors, like the companies who sell these collections, they do not make any guarantee that the cDNA version is full length, meaning it has extended all the way to the 5' end of the mRNA. It can very well be the last 300 nucleotides, 500 nucleotides towards the 3' end. So that is EST, expressed sequence tags. Now think about an experiment conducted in early 2000. I have done that experiment. So in those days, the genome sequences were still not fully known, and some of the other technologies, I will come to it in a minute, were not fully established. So in those days, how did people do microarray analysis? So the way they did it is just like the way I taught you how to do EST analysis, cDNA construction. They would make this EST library, which are basically the same as cDNA library, just that no guarantee that 5' end is represented. They will say your experimental system is human. Okay, so you will take cell types which is likely to express almost every mRNA that is possibly expressed in humans. Okay, so you can use fibroblast cells or embryonic stem cells, or your guess is just as good as mine. But you try to use a system that is not very specialized, so that you have a fair chance of getting a very broad representation of all the mRNAs that are possibly expressed in an organism. Please note, not in a specific cell or tissue type, but in a whole organism. Then, just like your cDNA library, they are all cloned in a plasmid, in a cloning vector, then transformed, and then they are plated. Okay, individual colonies, the way I showed it to you during cDNA library construction, individual colonies will be transferred to a multi-well plates. Okay, colony 1 will go to well 1 in plate 1, colony 2 will go to well 2 in plate 1, colony 3 will go to well 3 in plate 1, and so on and so forth. And you will eventually have, if you are talking about human genome, which typically is known to express 23,000 mRNAs, and because I explained to you about the overrepresentation of certain types of genes in the cell, like the housekeeping genes, the ribosomal protein coding mRNAs, the actins and tubulins and GAPDH. So to maximize the possibility that every mRNA is represented in your library at least once, you go typically 3 to 4 times the genome complexity. So if it is 23,000, you try to go, anywhere between 70 to 100,000 cDNAs. Those are arrayed in, 96 well plates or 384 well plates. Let's say 384 well plates, just for the sake of simplicity. Let's assume it to be 400. So you will have, 150 to 160 such plates to have a total collection of 70 to 80,000, like 60 to 80,000 clones, cDNA clones, which from each of these plates, these 384 well plates, can be like the bacteria can be transferred from each of these wells onto a culture plate, which will also have, like 96 well or 384 well. Generally, it is 96 well. You transfer it there and grow the bacteria. Okay, and then you grow the bacteria. You isolate the plasmid in there, following mini-prep protocol. It is just that instead of isolating plasmid from one colony at a time, one culture at a time, you are isolating plasmid from 96 cultures at a time. And then you take pure DNA and immobilize it on the glass slide, which will be chemically modified. So because you know exactly which DNA is being spotted where on the glass slide, this is where the advent of inkjet printing technology helped us to map these spots very specific, very precisely on a tiny glass slide, even when it has some 40-50,000 different spots of DNA. And you can have two such slides, and that is your microarray representing the whole genome. Okay, once you have that, then you isolate mRNA from condition A, label it with red, mRNA from condition B, label it with green, then you probe it on both the slides, and then you do image analysis and you figure out which of the genes that are exclusively expressed in condition A or in condition B. Now, please understand that all this time, till when you spotted those on the microarray and when you did the hybridization, you do not know which spot represents what cDNA. You have never sequenced it, okay? Because sequencing 100,000 clones before spotting them would have made a lab bankrupt in those days. So only when you get differential expression, that is those green or red spots, you go back to the original bacterial plates in your library and isolate plasmids from there for sequencing, and then you get to know which gene is differentially expressed under condition A or condition B. Okay. Now, that was the trend for at least 10-15 years before revolutionary new technology came up, that is called the oligonucleotide array. Now you need to understand that oligonucleotide array, advent of it, were dependent on several other discoveries that went before it. First is that by this time we knew the genome sequence of every major model organism that we work with. How is that helpful? So you think of an EST library and genes that are very close to each other in sequence, like say my favorite gene, BMP. There are 20 different BMP homologs in humans, okay? And chances are that many of them will have close sequence similarity. So when you get an hybridization with cDNA probe, it is not necessary that it gives you indication about a very specific homolog. It can be, I'm not saying it used to happen, but there is a possibility that you are recording expression of a homolog that and not the gene that you thought you were recording. So when the genome was sequenced, then it was very easy for scientists to conduct large-scale alignment of all the mRNAs. This is a purely theoretical exercise because you know that these are the 23,000 genes that are produced in humans. So you get the mRNA sequence of all those 23,000 and figure out a stretch of 60 nucleotides, 60 nucleotides that are very specific for each of these mRNAs. And that is not very difficult, okay? Even if not in the coding sequence, maybe in the UTR, you will find a stretch of 60 nucleotides that are unique for a particular mRNA. And the probability of two different mRNAs having identical 60 nucleotide stretch is very low, right? So now this is a pure theoretical exercise. And here you know which gene you are talking about. So for each of the genes, you can have an Excel spreadsheet, where gene 1, gene 2, gene 3, gene 23,000, and corresponding to that unique 60 nucleotide sequence. Then you can program an inkjet printer like device, it's a robot, which will deposit oligonucleotide like nucleotides just in the same sequence as is there on your Excel spreadsheet. But it will deposit the nucleotide in a given order for each of the 40, 50,000 spots on the glass microarray. So, I'm sorry, my stylus is not working. I'll fix it soon. But, just to give you an idea that, say gene 1 has a sequence of ATCG, it is going on spot 1, and gene 2 has a sequence of GGAT that is going to spot 3, then you can program it so that on spot 2, first A will be deposited, then T will be deposited, then C will be deposited, then G will be deposited, and in spot 3, first G will be deposited, then again another G will be deposited, then an A will be deposited, and so on. So you can program in which order nucleotides will be deposited on each of the 40,000, 50,000 spots, and the same chemistry that is used for DNA synthesis, the way primers are made, the chemical synthesis of DNA, the same chemistry will be used to actually construct the oligonucleotide of 60 nucleotide long, so or 60 Mars, they are called 60, the number is 60 hyphen MER, 60 Mars for each of the 40,000 spots will be created, will be synthesized simultaneously on the glass array. This is done purely robotically, and this is possible because of the advent of microlithography and solid phase DNA synthesis technologies, okay? And of course, sequencing of the genomes. So now, as opposed to the EST-based technology, now you know the precise sequence and location of each of the cDNAs on your microarray. Okay, so this is the fundamental difference between an EST array and an oligonucleotide array. In EST array, you do not necessarily know the sequence of the spots on the array. In oligonucleotide sequence, you definitely know the sequence of each of the spots on the array. And like here, representation is not a problem because in EST you had no clue what you were spotting, but here you know precisely what you were spotting. And you can go one step further, that you put certain spots that are not there at all in human genome but present, say, in yeast genome, or like certain genes in the human genome, you put 10 different spots for the same gene, like say my favorite gene BMP2, 10 different spots of BMP2, 10 different spots of GATA3 transcription factor, and so on and so forth. These are internal controls, because if the microarray hybridization worked well, then you will not expect any mRNA from human cells to hybridize with the yeast spots. Okay, so yeast spots will not light up. And if there is mRNA for BMP2 or GATA3, then all the 10 spots for BMP2 or all the 10 spots for GATA3 should light up. So these are called negative and positive controls, and the companies have developed algorithms by which they can assess how well hybridization, microarray hybridization worked, just by looking at how all the negative control spots behaved, how all those positive control spots behaved, and so on and so forth. Okay, so this is what it is. about the array. So what is there on the slide is a critical determinant. But nowadays, when you will be doing these kind of experiments, you will not perhaps worry about EST arrays at all. You will all be using oligonucleotide array. Then the other critical determinant is what is the probe? How is it obtained? And this is where your genius comes up. Okay, say you want to know the genes that are downstream of a signaling pathway. Okay, say GPCR signaling pathway. Okay, now GPCRs generally work with the first wave is through post-translational modification. But then there is another wave that is nuclear in nature where transcriptional changes also happen. Now, this is something that I want to emphasize, and I really would appreciate if you can imbibe this concept. Take it from me that after a signaling event, whether it is a GPCR signaling or BMP signaling or whatever, from the time the ligand binds to the receptor and transcriptional changes happen in the nucleus, then the mRNAs are transcribed, translated, the resultant proteins are folded. This whole process typically will not take more than an hour. So now, if you are looking at mRNA abundance after 5 hours, then what I am going to say next is a distinct possibility. That if you believe me for a minute that after a signaling event within an hour, the transcriptional events have already happened and the corresponding proteins are already made from the first wave, then is it not possible that in the first wave of genes that are expressed, meaning transcribed and translated, there will be some other transcription factors or signaling molecules? That is a possibility, right? That one signaling event promoted transcription of new kinds of transcription factors or signaling molecules, which in turn will influence the cell again to transcribe another set of genes. So therefore, depending on what time after you have manipulated the cell, you are looking at the transcriptome, you will perhaps see either the immediate early responsive genes, meaning genes that are truly transcribed dependent on the manipulation, or you will be seeing intermediate or late effect genes, which are most likely due to secondary or tertiary effect of the original set of transcripts that were made. Like I would alert you particularly to those kind of analysis where people would look at gene expression changes in cancerous cells after to identify which are the genes that are misregulated for cancer, they will compare cells or mRNAs from a cancerous tissue versus a normal tissue. Now, these kind of investigations have been done a lot, and I do not know of a lot of insightful information coming out of this. One of the reasons being that think how it is done. A cancer patient, his or her sample will come to a scientist if and only if the person reports to clinic. And clinic, the person will report to the clinic only when he or she would have symptom. Now, from the first event that caused the transformation, cancerous changes, to the time point when the person was symptomatic, perhaps is a month, 6 months, a year, couple of years afterwards. So do you think that the gene expression changes that are causative of cancer will be detectable after 2-3, like after such a long time, when the immediate gene expression happens within an hour's time? That may be one of the limitations why these studies did not provide a lot of insight. Okay. Same will be the case if you are trying to identify downstream targets of a signaling pathway or a transcription factor. For some strange reasons, many scientists, they will just treat the cells with a signaling molecule, signaling ligand, and will take the RNA after 24 hours. Why after 24 hours? What is the rationale? No answer. What instead should be done is logic should be applied to figure out when you expect the first wave of transcription to be done after the manipulation. Maybe few genes that are known to be transcribed due to the manipulation. So you check, you do the manipulation to the cell and then you check after 5 minutes, 10 minutes, 20 minutes, half an hour, 2 hours, that when do you see emergence of that known gene first. Once you see that, okay, that gene starts to be expressed around 30-minute time point and goes down after, say, 1.5 hours. So you know that between 30 minutes and 1.5 hours is the time frame when the first event of transcription is happening. So you take the cells that will be manipulated with your favorite transcription factor and the cells that will be fake manipulated. In each one of these experiments, you will have to have a control. So say if it is a transcription factor, you will transfect with a plasmid expressing the transcription factor, one cell, and the other cell will also be transfected, but it will be with an empty plasmid. So if transfection process by itself causes some gene expression changes, that will be constant in both the ones. If you get certain genes expressed only in the cells that were transfected with expression construct of the transcription factor and not in the other, those are the genes that you are interested in. Or much better is with signaling molecules. Okay? Because signaling molecules, you can buy the proteins as reagents from companies, and you simply add the protein to one cell, and on the other you simply add the buffer and not the actual protein, and you see the gene expression changes. And you can do it under temporal regulation, meaning you want to know what is the first wave of genes, the second wave of genes, and stuff like that. So therefore, in each of these time points, you will actually quickly harvest your RNA from the cells, and then you make cDNA. So therefore, how you are making the probe and how it is obtained will make a whole lot of difference. There are so many different scenarios by which you can which you can exploit to understand transcriptional regulation in the cell, that I will not be able to exhaustively describe each one of those to you. You should read online resources, papers, etc., to learn different applications of microarray. Then what is the control? That is a very, very important question. If you want to know what are the genes that are expressed only in heart tissue, cardiac muscles, okay? How would you know these are genes expressed only in the cardiac muscle and not in any other cell type? Which will be your reference cell type for cardiac muscle? Will it be skeletal muscles? Will it be, lung cells? Will it be bone cells? How do you determine which is a good control population? For others, it is fairly straightforward. If you want to know genes that are expressed due to presence of a specific transcription factor, you can take cells and express transcription factor in one cell and not in the other one. Signaling molecule, fairly straightforward. But if you are comparing tissues or disease cells versus normal cells, you have to think what should be a good control. And sometimes you might want to have multiple controls. Maybe if you want to know the genes that are only expressed in cardiac muscle, maybe you should use skeletal muscle, maybe you should use lung, maybe you should use skin, and see that what are the genes that are present only in the cardiac muscle and not in any of these cell types. Okay, I'm making it up. I'm not saying that this is exactly how you should do it, but all I'm trying to emphasize is that you have to think through what will be a good control. And you must remember that everything said and done, microarray experiment, the outcome, the output of microarray experiment is merely a suggestion. It is just a suggestion that these are the genes that are expressed preferentially under condition A or condition B. It is not written on stone. There are many reasons why microarray experiment may or may not reflect the reality. Therefore, you must have a good validation method. Now, in the literature, I often see that the validation method people use is quantitative RT-PCR. I'm sure it has its own merit, but this is also hybridization based process. Okay, so if the microarray result is skewed or is erroneous because of differential hybridization, depending on the oligonucleotide sequence, hybridization between the probe and the template can vary. Like, even though both the genes are expressed at the same level, just because of differential hybridization efficiency, one gene may appear to be more or less expressed than the other gene, simply because the hybridization was not as good or as weak as the other one. So using quantitative RT-PCR as a validation method, therefore, perhaps is not very appropriate, just because that is also a method dependent on hybridization in one sense. In our kind of work, developmental biology work, good validation is RNA in situ hybridization. So you actually say, I want to know the genes that are expressed only in developing bone and not anywhere else. So I design a microarray experiment, and then I get the suggestion from there. And then if I take sections of developing bone or sections of developing limb, and I see that the RNA in situ hybridization signal is present only in the bone and not in muscle or in cartilage or in tendons or anywhere else, then I know that this is a gene that is specifically expressed in the developing bone. Okay, or say I want to know genes that are expressed because of BMP signaling in bone. Okay, so I will use cell lines that are either experiencing BMP signaling or not. And it will be an inducible manner. Like, it is not that the cell has been experiencing BMP signaling for the last 5 months, like a standard knockout cell and a standard wild type cell. Then I will not get any answer. I will take the same cell and I will try to go as close to the in vivo cell type. If it is bone, then I will go for bone progenitor cells in vitro. If it is kidney, I will go for kidney progenitor cells in vitro, and then I will add BMP in one set, and I will not add BMP, just add the buffer in the other set. And after an hour, 2 hours, 3 hours, I will collect the RNA, make cDNA probes, and I will do the microarray, and I will see what are the genes that are likely to be differentially expressed. So I will get a suggestion, and perhaps I will pick top 100 based on the fold difference, and then conduct RNA in situ hybridization on actual developing limb to see which ones are expressed only in the bone. You can do the same thing with a signaling pathway respondent, or again, you have to use your own judgment for a good validation method. And then finally, the interpretation of the data, that whether what you are looking at is really an outcome of the manipulation that you have made, or it is there is some other possibility. See, one thing you must know today as you are entering a research arena, whether you are an MTech student or a PhD student, data is never wrong. Okay? Data is always right. It is the interpretation of data that's what makes or breaks a scientist. Okay, so when you are interpreting a data, you have to be very, very careful that the way you are interpreting, is that the only way to interpret it, or is there some other possibility? So that is something that you will have to learn yourself. So this is your textbook description. So in this specific example, the scientists wanted to know which are the genes that are expressed by fibroblasts. Fibroblasts are cells which do not have a specific shape. They are typically there in most of the tissue types. They proliferate a lot, and they respond to different kinds of signaling molecules and differentiate and become part of a tissue. Okay, so fibroblasts are treated with or without serum, and then they're conducted to then total RNA was isolated, and then in the process of making cDNA, they were treated with red dye or green dye, and you will have cDNA from fibroblasts without serum that are green tagged, cDNA from fibroblasts with serum that are red tagged. You mix them, you probe the array, and then do the image processing and figure out which genes are expressed only because of serum treatment, like the red spots here. So that's how microarray is done. Sometimes what you can do, to take it further, is to do clustering. What is clustering? Meaning, you take the samples at, so the specific example given here is that, say you take your cells and added BMP to it, and the control is where you do not add BMP, and you isolate mRNAs and make cDNA after 15 minutes, after half an hour, after 1 hour, after 3 hours, and so on and so forth. And you see that which are the genes that are coming up very early on. Okay, here you can see that this A and C are two clusters where after a certain time these two clusters of genes expression went up, while expression of certain other clusters, like those that are in red, they went down. And this cluster D, they went down first, soon after treatment with the growth factor. Or this cluster, they went down, went up immediately after treatment with the growth factor. So these are multiple different arrays, and you are trying to look for their common behavior, and then those that are behaving in a particular manner, you cluster them. Okay? That's what is cluster analysis. And this just infuses the power of your prediction through microarray analysis. Okay, so you can identify co-regulated genes this way. Co-regulated meaning genes that are expressed in the same place at the same time, they are likely to be co-regulated. And you will understand this concept a whole lot more when I talk about transcription and transcriptional regulation. Right now, please remember this term co-regulated. Now, let me give you some final comments. First of all, if you want to know whether a particular mRNA is present or absent in a cell, please do not propose microarray experiments. There are much easier ways of doing that. In the olden days, people will be doing northern hybridization. But nowadays, you can easily do RT-PCR, reverse transcription PCR, If the mRNA is expressed, you will get a PCR product. If the mRNA is not expressed, you will not get a PCR product. If you want to know whether a particular mRNA is produced in a particular cell type in vitro, you can do RT-PCR. If you want to know if a particular mRNA is produced in a particular cell type in vivo, I would prefer that you do not dissect the tissue, you do not destroy the architecture, you simply conduct an RNA, like you simply make a whole mount or a section of the tissue, and then you conduct RNA in situ hybridization. To know the power of RNA in situ hybridization is that you get to know the spatial and temporal expression pattern without destroying the context. But whenever you are talking about a whole group of genes, and you do not know which group it is, that is the most important part. A group of genes and you do not know which group it is, then microarray is a very good experimental approach. RNA-seq does exactly the same thing as microarray. The technology there, if you remember next generation genome sequencing that I taught you earlier, is the same thing. You simply isolate mRNA and you do the cDNA synthesis, and in the process you get the sequencing, right? Just like the next gen sequencing, but you are doing it massively parallel ways, the way it is done for next generation sequencing, whether it is genome or mRNA template for cDNA, it doesn't really matter. You get it. RNA-seq is the preferred method by many. It is still a little bit more expensive than microarray, but it is still preferred simply because microarray, one variability comes from the efficiency of hybridization. Like as I explained earlier, that just because you are getting a very strong signal for a spot does not necessarily mean that that mRNA is more abundant than the next spot, which is a much lower signal, because it is possible that the particular mRNA has a very high or very low GC content. If the GC content is very low, then the hybridization efficiency will not be very high, and it may artificially look like that gene is expressed less. But when you were doing RNA-seq, there you are actually making cDNA out of the mRNA. Therefore, it is highly quantitative. You can actually count the number of mRNAs in a specific sample. Okay, so just remember that much, that RNA-seq is much more quantitative than microarray, but RNA-seq is a little bit more expensive than microarray. So depending on which lab you are in, how critical is the information about differential expression for your further experimentation. Please note that just knowing differential expression doesn't do much. You, you want to know the differential expression for your subsequent experiments, that is, to know why these genes are differentially expressed, what advantage the organism gets by expressing a certain set of genes in one condition versus the other. So these are functional analysis. So those are the actual reasons why you are doing differential gene expression. So depending on how critical it is for you to know the differential expression accurately, you may want to choose microarray versus RNA-seq, but they give the very similar kind of output. And both are nonetheless suggestions. You should follow it up with the validation techniques before you, particularly if you plan to move on to do functional analysis, then it is not at all advisable before you have done some kind of validation method. Okay, So that is all that I had to talk to you about microarray techniques. After the quiz on October 3 and 4, we will learn the new topic of protein expression in biotechnology. Okay, so that is all. This lecture will be part of the quiz on October 3, 4. Therefore, please listen to it several times and refer to online resources. And of course, you should feel free to ask as many questions as you would like in the next two discussion classes tomorrow and on Friday. And if we can manage another on Thursday, perhaps. Okay. With that, I will end here and thank you very much for your attention.